Okay, good evening, everyone. This is Sue from New Jersey Mycological Association, and this is Taxonomy Tuesday, where members share their fungal finds, and we get together and see them and go through the identification to confirm or dispute anything, or just to help novices learn the first easy ones. We first go to anyone who wants to share from their computer. If you do, if you just type your name into the chat. Otherwise, we would go to the emails. Okay. So let me go to the emails. Joseph. Let me share my computer. Can everybody see my screen?
Yep, I can see it.
Oh, yeah.
Yep, see it.
Oops. It's here. I have to close that. Open the window. Okay.
Oh, look at
So
that.
here's the gills.
Could have had like a like a lavender shade to the gills.
Right. Have you heard of bluets?
I have heard of them. Yeah, I have heard of them. Yep.
Is that a
look-
ring zone on the stalk?
Is that a what?
A ring zone on the stalk, or is it just some dirt or something?
I think there's another. I didn't see a ring on the stalk of the thing.
Yeah, sometimes this is okay. Yeah, get a close up here. Oh yeah, I don't see any
Yeah, it was just where it was caught. It looked like, right? It looked
Looks like
it,
it's turned to a different color, yes.
yeah.
now the color of the cap's a little bit pale for bluets.
Yeah, we've had a lot of
And,
pale ones this year.
Yeah. Oh yeah. Okay. Yeah. Well, they might be Lepista irina. But the other possibility here is I'm just looking at the really flaky um, uh, upper part of the stalk. That reminds me of hebeloma, which are poisonous. But true. The rest of the mushroom looks more like a lepista. So
Did we take a spore print of this?
I haven't yet. No, I haven't yet. Oh, let me see
Would
here.
that help, Dave, between the Lepista and
yes,
the
you need a substantial print. Okay, a
Okay.
heavy enough print. But the hebeloma is going to be light brown, decidedly light brown.
Uh-huh.
The Lepista, whether it's Lepista nuda or Lepista irina, and by the way, those species are now currently housed in genus Collybia. I'm not going to get into that. It's kind of ridiculous, actually. Hope, I talked to Noah Siegel about it. He said they're going to fix it. We'll see. But I still call them Lepista or Clitocybe, nuda, irina. So Lepista irina is like a bluet, like a Lepista nuda, but it's usually
Right.
a lot paler. But Lepistas have spore prints that are not that different from white. So it's slightly pinkish, sort of fleshy, pinkish. You get a nice heavy print on a white, non absorbent surface. If you take your print on paper, or paper plate, or something like that, the paper is going to draw moisture out of the fruit body. It's going to affect what the print looks like. So
Okay.
you want some sort of non-absorbent surface. You can get these plastic picnic plates like at um, discount places. Actually, you can get them pretty cheap. So perhaps like a white plate and a black plate.
Right.
But when you're looking for contrast between, when you're looking for a print to be not white but barely not white, then you want to take the print on white because you have a better chance of seeing the contrast.
Right.
But yes, a hebeloma would have a decidedly brown print.
All right. How long do you have to take the spore print for? How long does it take?
It depends. Sometimes you put a mushroom out for 5 or 6 hours and you have a nice thick print.
Okay.
Sometimes you look after all 24 hours and there's still not much there.
Okay.
It depends. These look pretty fresh. You should be able to get a good print out of these in 10 or 12 hours.
What
Okay.
about the habitat? I see this Lepista irina, which I'm not familiar with, says is associated with beach. Well, what were these mushrooms near?
Well, it seemed like oak trees were right there. There's acorns all over the ground and a lot of oaks.
Okay.
Yeah, Lepista mushrooms are irina and nuda are saprobes that that decompose lots of different types of like forest or lawn waste. So they can be either one can be found in a variety of settings. Although Lepista irina tends to be more of a forest mushroom, so maybe beach. I mean, honestly, the places where I have found what I identified as Lepista irina have been in places where there's beach. So
Okay.
that makes sense to me.
Well, now that I know there's poisonous lookalikes, I won't eat this until I do a spore print.
Yeah, I would try to get a print on this first.
Okay.
Now
What
Okay.
about
really
the odor?
looks more like Lepista to
It
me.
almost, yeah, it kind of had like, I did break a little piece off. It had like maybe a sweetish smell, like a little.
Oh, that's a good sign. Yeah, the Lepistas have like are sort of aromatic.
Yeah,
They
it
have like
did.
a musky,
Yeah,
pleasant odor.
it almost smelled a little bit like an orange or something.
Yeah. Um, there's a lot of different Hebeloma species, but most of the big ones that look kind of like this will smell like radishes.
Okay.
But you know what? I'm looking more closely here at this photo that Sue is just allowed to stay up. And the top of the stipe doesn't look so much flaky as it looks just a little bit very fine scales.
Yeah.
So I don't think this is Hebeloma, but it's probably a good idea. I mean, just in general, these sorts of things are a good thing to learn to do if you're just
Sure.
if you're just learning to identify some of these mushrooms that you want to eat.
Sure.
You should go through these sort of exercises.
Sure. Thank you very much.
Could someone put that in the chat, the name?
What's that?
Could the name be put in the chat?
Yeah, if I can find the shows here. Oh,
Okay.
chat. Which name do you want? The Lepista irina?
Yeah, at the spelling on that, the whole thing.
I think I just put it in.
It's
I
an
think that's how. I
Well,
think
you
that's how
had a
you
typo,
spell
but
it.
yes.
I don't think I'm missing any vowels. I think
Thank you.
that's how it's spelled.
Thank you so
If
much.
not, it's close enough that if you Google it, you'll probably get it.
Yeah, I was not getting it because I was not spelling it right. So thank you.
Oh, sure. Or Collybia nuda. Yeah, Collybia is the new genus for these. I've been told that might not last all that long.
Yeah.
Yeah, well, thank you, Joe, for sharing. Is this your
Oh,
first
oh.
time on Taxonomy Tuesday?
Yes, it is.
Okay, well, I hope you
Mm.
join us again. I know it's sort of the end of the season.
Yeah.
But these Lepista has been coming out for a few weeks now, so maybe they'll
Yeah.
go for at least
Sounds
another
good.
week.
Thanks very much.
Oh, so you're getting those in New Jersey. I have seen hardly any of these this year
Me too.
Yeah.
up here
Yeah.
in Pennsylvania.
Yeah. Way down in South Jersey.
Yeah. Oh, you know what? There was a lot of rain in early October, I think, or late September. That
Mhm.
probably allowed the mycelium to grow. These are a saprobic species, and I think the mycelium has to sort of start from scratch almost every year.
Hmm.
Hey Joe, the only other thing, it was a nice set of photos for identification, by the way.
Oh, thank
Yeah,
you.
yeah, I
And
agree. There were good details to see there.
and
Great,
and
great.
and popping the whole bottom out of the ground is a lot of times helpful in identification.
Okay,
Yeah,
Yeah.
and
that's good
especially
to
with
know.
the Lepista, because there's a nice mycelial mass, and it's not really, it's barely hanging on
Oh,
to anything. So, but the mycelium's like stuck to the leaf litter generally with those, so that can really help identify
okay. Okay, so try to try to have a little longer stem all the way to the bottom, sort of
all
thing.
the way to the bottom. Yeah.
Yeah, got it.
Yeah, and he even got the smell, which is really good.
Yeah, well, my daughter said that's what it might be, and she said they have a sweeter smell. So I did. That's the only reason I smelled it, because she knows a little bit more. She lives in another state, but she knows a little bit more than I do about this stuff.
Yeah, I think this is a Lepista, but it's always good to, like I said, just go through the exercise
Right.
of,
I'll definitely do that.
yeah.
There was like 8 of these things right together. It would have made a nice meal, a nice few meals.
Well, you could put them in the refrigerator, and um, a friend of mine who's a mushroom enthusiast and a trained chef told me a trick for keeping fresh mushrooms in the refrigerator for a few days, and the trick is this: you put them in a deep porcelain or glass bowl, and
Okay.
you drape over the top of the bowl a moistened paper towel.
Oh, okay.
And then if you're keeping them for more than a day, you re-moisten the paper towel.
Okay.
And I
That's good to
have
know.
done this, and it works pretty well.
That's good to know.
Thank you, Joe.
Thank you.
Hunter, you're the next one that came in?
Oh, okay. So I went to the Cultivation Curiosities last Wednesday, and the topic
Oh.
was dye mushrooms and cultivating them. But the blue morph of the turkey tail came up, and Emily was wondering if it's a dyer, as it is listed, I think, on the NAMA site as one. But so I did see it. I'm seeing it. I saw it in a couple different spots today in Northern Illinois, and so I decided to take a picture of it. And then I also, for Emily's sake, I did ask on the Dyer's Facebook page, the lichen and you know what I'm talking about, the Facebook group, Alyssa's group, if people thought it was a good dyer. And it was, I only got 2 responses, but they were both were no. And then I did talk to someone else who I know dyes, and he said that you really need a lot of it to get much of anything, but he was going to experiment with it a little bit more. So yeah, and I have another picture of it, the other group of it, one down, yeah, that one, and that was really pretty too. So I just thought I'd share that.
Yeah, I
And
appreciate it because I don't think I've ever seen the blue ones.
oh, okay, yeah, and I have, and apparently they're coming out right now. So I'll look for more, but it doesn't seem, I mean, right now, it's not too promising from what I can tell as a dyer.
Well,
But
considering they're paper thin, you can imagine you'd have to collect an awful lot.
yes, yeah, exactly.
Did the bluer one seem to grow on anything specific or different that you know of?
It's a good question. Could be conifer, but I mean, I don't know. I probably not. I would say they might grow on something different, but I think I don't know. But I mean, both of these were growing on pretty decayed wood, and it could have been a variety of things. I'll try to pay a little more attention. I'm thinking maybe a dead ash, maybe dead elm, maybe probably these weren't growing on oak, but there is a lot of white pine in that area. But you can see deciduous leaves like around this one. So yeah, probably yeah, it could be.
I mean, different trees just pick up, like all plants, they just pick up different minerals or something. So maybe if they're growing on,
Could be
specific.
cottonwood even. So I know there were some cottonwoods around. Yeah, no, I know. It would be nice to know, and pay more attention. So I will. Maybe I'll have something to say on that next time.
So the last few years, I've been seeing a lot of turkey tail on the ash trees that have been dying here for the last 15 years or so. And I'd have to look through my photos, but I think a few of them have looked kind of similar to this.
Yeah, and then it could be maple. I mean, I know the brown ones will grow on maple. So I don't know.
I think they're not too picky in general, but I believe that they're supposedly only grow on hardwood.
Oh, okay. No
I'm
comment.
not sure about that. I'd have to look it up, but I think.
Yeah, I mean, I don't feel like I've seen it on conifers, but I don't know. I'm gonna look right now though. Let's see. Quo says hardwoods rarely on the wood of conifers. Annual, and that's it.
Rarely doesn't mean never.
Nope, it doesn't. So it could be. But yeah. So and then I found yeah, that's good. Hygrophoropsis or aurantiaca. And I'm just surprised. I've shown it before and we've seen it before. This is the false chanterelle. I'm just surprised to be seeing it so late. And I've seen it, and maybe I shouldn't be, but I've seen it a couple times in the last 2 weeks, 3 weeks, and each time I've been surprised. For some reason, I consider it more when the chanterelles are out. So I don't know if it's normal or it's
Normal.
Okay.
It's a light species. I mean,
Oh.
you can find it in the summer, but it's mostly grows in the fall. Was this under white pine? Are those white pine needles?
Yes.
Yeah, we had a foray here in Northeast PA last Sunday, and I bet you we saw at least 100 of these.
Oh, okay.
The needle duff under white pines. There were an awful lot of them, and some of them are orange, and some of them are pale, and some of them are kind of yellow. But they always have these repeatedly forked gills.
Yes.
And that is why they gained the common name false chanterelle, because that is a trait associated with Cantharellus, the repeatedly forked gills.
Yep, and they're also somewhat decurrent.
Yes, and decurrent. Yes, and decurrent, and the color is not altogether different as well.
Right.
Yep.
White spore print.
White spore print. Yep.
But they don't they don't come up singly like chanterelles do. They tend to be clustered.
They come up singly. They come up in.
They don't have to be, but
they grow all different ways, really.
Yeah, and sometimes like out of decayed wood.
Sometimes on decayed wood.
But chanterelles wouldn't be doing either of that.
Yeah.
Yes, true chanterelles are always terrestrial.
Right.
But these guys are too, a lot of times. I mean, not always, but a lot of times they will. And sometimes they grow singularly. And chanterelles sometimes grow in clusters. I found clusters of smooth chanterelles
Me too. Yeah,
uncommonly.
right. It's not a rule, but it's most of the time they're solitary. Yeah. Yeah, so I just thought I'd share it. It's about the most colorful thing out there right now where I live. And I'm spending more time in the few sections of pine that we have. We have mostly deciduous forests here, so perhaps that's why I'm finding it so unusual, because I wasn't seeing it this time of year in the more deciduous areas. So yeah, I got that, and then a slime mold, which I think I showed the same one, and like really recently. I mean, it's a different picture of the exact same one. This picture's a little magnified, and then there's one where it's not, and yeah, that one. And I don't even, I don't, I Who's trying to speak? Please say something.
I couldn't tell if this is like a gala or something else. What did it turn out to be?
Oh, I think it's a slime mold, but I don't know what I don't.
Right,
Yeah,
just
I
like
don't think
Ogala,
this is
slime
like
mold.
Ogala,
It's
the
kind of
sizes
small,
of
but
the
I
fruit
can't.
bodies. Oh,
What
I'm
about-
sorry.
What about salmon eggs?
Yeah,
It says slime molds.
Hemitrichia.
Yeah,
Yeah.
trichia,
Okay.
hemitrichia, something like that, probably.
Yeah,
The sizes of
not
these
like-
fruit bodies are too variable for like Ogala.
Yeah, no way is it like Kogawa. No, right. Yeah, so maybe Trichia, what, decipiens? Maybe.
They're
No.
hard to separate. Even trichia versus hemitrichia, I believe, is tricky.
Oh, okay. Salmon eggs is Hemitrichia.
So these
Okay.
don't have any stalk of any kind? They're just a blob?
Well, the trichias have a little stalk, and then but I didn't capture that in my photo at all.
I think they often start off stalkless when they're young, and then as they mature, the stalk kind of elongates.
Okay.
Yeah,
Mhm.
which makes it even trickier. You know what? Hunter, do you carry around a loop with you?
Yeah, I do. I
You know
haven't.
what? If you find something like this, see if you can take a photo of just like one of them through the loop. Sometimes it works, and
Okay.
then you'll capture some of the subtle detail. Possibly, it's worth a try. You know, sometimes it works, sometimes it doesn't.
Thank you. Thank you for that encouragement. Yes, I need continued encouragement, and I will try to remember to do that. Thank you.
Thank you, Hunter. Especially for the blue turkey tails. Yes, so I get to see those.
Good.
Okay, next. Dave, is not yet 8 o'clock, so shall we go to Brandon?
Yeah, go to Brandon first. So just in case, you know, we don't want to run the risk of having the Jeopardy theme recorded on
Okay.
our.
All right, so these first two are from the 4A this past Sunday at Estelle Manor. I'm using this name lacmus, pretty much based on the color of the gills. Even when they're young, they have a tinge of violet, and then that mature one is kind of blue-gray.
Beautiful.
Yeah, really cool.
So there's another species name that pops up when lacmus is proposed. At least it used to on Mushroom Observer. I forget, but it's a name that suggests, you know, it comes from the purplish tinge of the gills.
Something like purpureus or something.
Yeah, it's not
like
You know what?
that.
I'm gonna find it. I'm gonna find it now.
Is it
Um,
subviolentis?
Subviolentis. Yes, indeed. Thank you. That's it. Yeah. So there's always like some arguments over whether these are lacmus or subviolentis.
But they're
But
all in
you're,
the cup.
you're certainly on the right track here.
So it's in the same family. Genera.
Yeah, Cuphophyllus.
Cool.
Uh,
And it was
subviolentis.
subviolentis. Yeah.
Violentis. I'm sure I'm spelling that wrong. Violentis.
But these, you know, the wax cap group, these things fit right in there. You know, have a more of a greasy than a slimy cap, but has that Cuphophyllus look with the decurrent gills and everything.
And was this found in Spagman or Spagman? Spagman?
No.
What is this?
Well, there should be probably an in situ photo.
Yeah, I'm looking at it. I don't know what that is. It doesn't
That's
look
not
like
in it.
Spagman. That's
That's
not
not
it?
an in situ. That's not in situ.
Yeah, I only got those three.
Shoot. No, it wasn't in moss. It was actually at the base of a couple of the few sweet gum trees.
Okay.
Just kind of in the debris, you know. But yeah, I just
Nice.
staged them on moss just so you can see the colors better.
I always do that, and then people are like, "Was it growing on moss?" I'm like, "Actually, no."
Yeah, if you look at it, they're not growing right there.
But is it
Right,
good?
they just yeah, they're
The
not growing.
last photo that was on the bottom of the view, you can see that it looks like they were attached to soil. And I think these are mainly terrestrial. I haven't found this species in quite a while, whichever of the two it is.
Right. Yeah, I'll look into the subviolentis, but if it's just an argument between the two names, I'm just going to call it lacmus. And
Yeah, I
call it a
think
day.
it might be. I think it might just be a disagreement on which name should be used. But you know, check it out.
Yeah, cool. Thank you. And then I found a Tolypocladium, which I've been seeing these posted recently, so I was really happy to find my first one of the season. So this is a parasitic fungus that parasitizes truffle-like mushroom. Lamphomyces is the genus on the truffle-like thing, which is underground. So you got to excavate these things and hope you get the actual host fungus.
Wow,
Is
that
this
was
the
cool.
is this the top or the bottom of that stalk? Is this
Top.
That's the top. It is up.
It's
Yeah,
blue.
so that's the
Oh, there it is. Okay, now I can see that.
that's the fertile surface. That's the
Yeah.
part where the spores are produced and released.
Did you get spores from this?
Yeah. Yep. So Dave, before we go on to the spore photo, what species would you say this is between?
Well, it's probably either capitatum or Longus segmentus.
Yep, that's what I went in there. I was like, and I know the difference. The spore dimensions are not ambiguous, so as long as I get some spores, I know I can ID this thing.
Yeah,
Then
I have managed to ID a few of these over the years just by lying the cap down on a slide. And the spores are actually a whole bunch of spores like connected together in this septate arrangement, but it's how long the thing is. You know, Longus segmentus, of course, has these configurations of spores that are really, really long. I forget, you know, the 80 microns or something. I forget. And capitatum is it's shorter. It's like 20 microns, maybe. I forget. I'm just guessing.
you're in the ballpark. Yeah.
Sand. Was that
What was that?
sand in the picture? Is it like a sandy area?
It's Pine Barrens-esque. So yeah, it's going to be acidic, sandy soil.
Okay. Oh, wow. Look at those grains of sand. That's so interesting to me. Neat.
Okay, and then if we go to the spores, and I have dimensions in the notes, the spore parts for these, I think, average somewhere around 5.5 microns.
Yeah, is there a difference between longosegmentum and capitatum in terms of the individual spore segments?
Well,
Oh,
these are the-
there's a good photo. This photo looks good. That last one.
well, you got to understand with those, yes, they're segments, but those segments are large. So when they're talking about the segments, this, well, they're not really segmented on longosegmentatum. They don't break apart. Put it that way. These
Oh.
break apart. So you see those little, they look like Tic Tacs, a line of Tic Tacs.
Tic Tacs, right.
And the long strain, you can see the individual one just above it, right?
Yeah.
That broke apart from the rest of its segmented strand.
Right.
Which is a thing that occurs with Tolypocladium fractum. Then there's
that's
this
right.
one.
There's that other one. I forgot there's a third one.
Well, the third one on Myco Quebec is I think it's valiform. And this fits that description to a T.
Oh, wow. Good.
So if we can cycle through these photos, I think there's one. Yeah, just keep going. Next one. Yeah, this is just a bunch of those spore parts, they call them. Yeah, this photo. So this is at 100 times magnification. And you see those tentacle-like things? Those are actually the asci.
So these long strings of segments are embedded inside these asci. How many of these strings are embedded in one of those?
If we go to the next photo, it's at a higher magnification.
Oh, look at that.
Wow.
Oh, nice.
I would guess, yeah, right? Isn't that wild?
Wow. Nice photo. Yeah. It's hard to tell.
You.
There might be some hiding behind these guys, but
at least
like 3.
I see at least 3, which would indicate there's probably a fourth.
I was going to suggest there's probably a fourth because it's almost always a power of 2.
Right. Yeah. So I'm thinking there's probably 4 of those in there.
Yeah.
I
But yeah,
mean, the vertical
and these
things.
Yeah,
Vertical rows. Okay.
Correct. And those asci, they are 200 microns. They're huge.
You probably get some good information from the size of the asci, actually.
Right, because with the other ones, they're more like a normal ascomycete. You know what I mean? Where there's 8 of those segmented spores within a normal looking ascus, right? So yeah, this one's kind of an outlier for the genus.
Oh well, that's great. You're able to nail this one.
Well, no, yes and no, because I've called something similar fractum before, but I can't get a good description on that one. It's just the only name that's available on iNaturalist that indicates, and I read a description one time that said it has these short spore parts, they call them, not segments. They call them spore parts when they break apart. But the description for valiform on Myco Quebec, which is not available on iNaturalist, that's the one that matches this description pretty much perfectly. So I don't know which one to call it. It's probably one of the two.
I think some of the names on iNat, quite frankly, are generated by The people who are doing a lot of sequencing, they're getting a sequence that doesn't match some of the more common names, and they're just looking through the literature and trying to find something to call the sequence, and then it just catches on and everybody starts using it.
And then,
The name.
yeah,
I think that happens once in a while on INET.
So how did you get this? Did you cut a piece?
yeah. Just, I mean, you know, a sliver of a sliver of a sliver, as tiny of a bit I could get, and then smash the ever living out of it.
Yeah, because I'm pretty sure the spores come like rocketing out of these things. I had one laid down in a container, and just on the way home, it looked like a gunshot in there. Like it was on the top of the container, the sides of the container.
Yeah, they're not shy about dropping their spores. But cool, let's go ahead and move on.
So I'm reading that fractum has a stipe that's less than a centimeter wide. Do you read that?
Where
Well, it says stipe slender, less than 1 millimeter thick. So what is the stipe then? It's not the
1
stalk.
millimeter?
Yeah, that's the couplet in that separates fractum from valiform in the Mingyu paper.
In the Ju paper? Is that what you said?
Yeah.
Yeah.
Couplet 24.
Oh, cool. Yeah, I haven't even hit that for
23.
this. I've been busy, but that's something I definitely need to look into. So it says for it's fractum, right?
Yeah.
The stipe is less than 1 millimeter.
Says stipe slender, less than 1 millimeter thick, and then for valiform it says stipe thick, columnar, 1 to 6 millimeters thick.
Huh. This is probably bigger than 6 millimeters.
Yeah. So maybe I don't know what they mean by stipe then. Maybe there's some other structure that
Yeah, because 1
I
millimeter.
think you're seeing this correctly. These things have heads. You know, they have caps. And so what you see below the cap is probably what's being called the stipe.
I
And
would think
so
so.
this valiform thing or fractum thing has a really thin stipe. That's interesting. I wonder if, you know, that might be a misprint. It might supposedly be centimeter.
could be centimeters.
But the other one's thick. It says the other one's thicker, but it's only up to 6 millimeters. Okay, right. So I'm sorry. So I'm wrong. Well, these just might be a little wider than they're supposed to be. That happens.
Yeah, I'm just going to the Myco Quebec page for valiform real quick. See
Yeah,
see what they said. So shoot. Yeah, I mean 0.5 to 1 centimeter. So
Well, there you go. That
yeah,
looks like about a centimeter.
yeah, probably a little less than 1.
Yeah,
So
maybe 1 centimeter. Right.
All right, cool. Yeah, I felt good about valiform for this thing, but the name's not available on INET, so
Yeah, there might be some misidentified things on INET. But or else you found something that is quite unique, you know, that's hardly ever found. And since it comes from the Pine Barrens, hey,
yeah, I happen to think that's kind of what's going on here.
Yeah.
Because Myco Quebec also says very rare for valiform. So probably just an under misunderstood genus in general, to be honest.
yeah, well, you might get the first post on INAT for this, which would, or at least in North America, let's say. So it's not on INAT at all?
Now the name's not available.
Wow. Well, that's pretty cool. You can be the first one because I think you've got a pretty good case here.
Yeah, I think so too. And we found it at a 4A, so
Did
Well, I can't-
any material get preserved?
yeah, I dried three specimens.
You know what? Steve Russell will probably sequence this for you for free because he'll do things that if you can make a case for something probably not being in his database, he'll just do it.
Yeah, I'm hoping to get someone on board with getting this one done. A
Yeah,
good
certainly.
priority sequence,
Yeah.
But all right,
Did we
cool.
sequence the truffle-like thing that it's growing on too, or do we know what it grew from?
I know it's a Lamphomyces. I have it between two species right now. I haven't. That one's a little bit on the back burner.
And I'm just wondering if they go together when you say that this grew out, they sort of want them together for sequencing or not. So, okay. Moving on.
Yeah, and I don't even have a good genus for this one. Some kind of coral fungus grown with hardwoods mostly. I mean, there's a pitch pine every 20 feet at this place. So
Is that really how you found that thing? Because I saw it on the table.
yeah,
Oh wow. That's a
So is that
pretty
on
thick pile of
soil?
duff there.
On soil?
yes, it was on kind of a sandy mound, like a sandy ridge, more or less. But yeah, not on wood.
Yeah, I've been seeing some posts. I forget if either on Mushroom Observer or INAT, I forget of some weird Ramariopsis species that I had not heard of before.
So do you know if Ramariopsis, because I hit this with iron salts, and it was negative for iron salts. It did not go green.
Yeah,
Which
so well, you've sort of eliminated Ramaria, I guess.
but just Ramaria, does that also
I
eliminate
don't know.
Ramaria?
I don't know.
Yeah.
I don't know.
Yeah.
It doesn't look like a Ramaria, though, honestly.
Not really.
What's that thing, Clavulinopsis or Clavulina? I always mix them up. Rugosa. That's small, though, and doesn't
Yeah.
cluster like this.
The
I
leader
got
in
a
the
book
club,
of those.
the leader in the clubhouse right now is I think it's Phaeoclavulina.
Oh, Phaeoclavulina. Yeah, those are kind of, they're not real fragile.
Oh, really? This was very fragile.
Yeah, that's what it looks like to me. And those Phaeoclavulina, they sort of look like Ramaria. Oh, and I think if you put KOH on those, you get a reaction.
Hmm.
With the what is it? Phaeoclavulina? I think it is, right? Yeah.
Some variation of Clavulina,
Yes, I think
Clavulinopsis.
it's Phaeoclavulina. Yeah, and they can grow in great big clusters too. I mean, really, really big clusters. And this doesn't really look like what I think Phaeoclavulina looks like.
Yeah, and I'm very unfamiliar with that one, so I could
Well,
be.
I think
totally
I
wrong.
think I know a little bit about it, but I'm not sure because this whole coral thing has just got to be pretty confusing.
Clavaria longicalis or something like it.
Bavaria longicalis. Bavaria long.
Longicaulus.
Yeah, never heard of it. Can we go back to the apex of this, just the tips.
Well, this one or the different one? This one?
Just any of them that can see the tips properly. Very blunt. You know what I mean? A lot of the names that were thrown around kind of get shot down with how blunt these tips are. I think. But yeah, that was something I noticed right away was they're very blunt.
Yeah, that's what made me think of these clavarias in this because a lot of them have that look.
These kind of things don't have any smell, right? I've never been able to smell anything from these guys. Do any of the corals have odors?
Some do. This one did not. But yeah, then I got some spores out of it, so I at least have that to kind of double back on. They're not very uh, extraordinary. They do have a central guttural, but I think a lot of these spores in these closely related genera can look pretty similar. They're small. There's that too.
Any kind of ornamentation there?
Yeah,
Maybe.
narrowly cylindrical, little bit of a bend to them in some profiles. But I think that that first photo where you can see it from the top, yeah, not too much to say about it more than other than ellipsoid.
Yeah.
Well, Ramariopsis kunzii, which is probably the most common Ramariopsis, has spores that I think are small and sort of roundish. So,
Yeah. And I found that the previous week, but it has more of the tips aren't as blunt as what we see here. And that, of
yeah.
course, is on wood, and this is not.
Oh, Ramariopsis kunzii is terrestrial.
That's true. I think I found, well, maybe you're right. I mean, I thought I found mine stuck to a piece of wood, but maybe
Well,
I'm
might have been
mistaken.
stuck to wood, but it's primarily terrestrial. It's probably fruits from wood in the ground, though. I don't think it's mycorrhizal. These
Yeah.
spores are kind of, look at the Q on them. It's probably like over 2.5.
A lot of these clavaria, I'm looking in the back of this book too. They have almost like a little apiculus, and then seems that many of them have a guttural.
Yeah, I think you can probably make out the apiculus on some of these.
Yeah.
Even the ones that are out of focus, you can almost see the apiculus better.
A lot of these Bisidia look the same too. If you scraped off a chunk, they're kind of oddly enough, like they seem to be in clumps, like a lot like the larger fruiting body, so like almost fractal in nature,
That's
which
cool.
is super bizarre.
All right, cool. Well, it was my turn to share a white coral that we just kind of talk about and don't really get to a species, but good stuff to actually look into. So appreciate that.
Was it
Then
easier?
I found this one today, and I'm calling it persicina, but I really don't know how to tell the difference between that and chrysoblema other than color. But as I'm learning with chrysoblema, that color can be highly variable. So
Does he go on? Because he pointed out to me, because I found one that I thought was Piscina, even though the color was not peachy enough, that it was the base that determined that for him, that it was
Yeah, this has a
chrysoblema.
lot of ornamentation on the base. A lot of looks like UV rings on the base,
I don't have a straight.
which
base.
would point towards
Oh, what is that?
chrysoblema or
Gasoi. There you go.
Muscaria variegasoia. Oh, look in the under the cap again. Is that partial veil not expanded?
No,
Oh, so you know what? I think persicina, is that the way you say it? I think that one has like a rather a fleeting partial veil. This one looks like it's going to be pretty substantial. I mean, that's just guessing, but the bottom of the stalk looks like it's got too much going on.
so the persicina, however you say it, doesn't have the quote unquote rolled sock thing
No,
going
it's not a
on.
rolled sock. That's a different thing. The rolled sock thing is like you don't have these concentric deposits. You have there's just like one rim, one really abrupt rim on the rolled sock ones. So those are the pantheroid ones, series Pantherinae.
Oh,
But this has the universal veil is formed these concentric rings on the base of the stalk. That's a Muscaria thing. But let's see. I'm pretty sure persicina doesn't do that. That it's more or less smooth. Let me look. I'm gonna look.
They're gonna be on Micro Quebec, Piscina, is that?
Nah, it's a southern species, so they probably don't even have it. Look on Mushroom Expert.
No, they don't. Yeah, I don't think Mushroom Expert keeps up with a lot of this stuff.
Mushroom Expert probably still calls it Muscaria variety persicina.
Yep.
Yeah, there's your stipe base. See, it's more or less there's not much going on the bulb.
yep.
Doesn't have the rings or.
The one in
Yeah,
the
like
middle,
the
though, on the bottom middle, looks like it's got a fairly substantial partial veil. It's hard to tell. The angle's not right. That's just breaking apart. I mean, that does look a little bit like Brandon's. And look on the lower right, that's got some deposits on the bottom. I'm not sure how else you would tell. I'm not sure what else you can go on. Those are persicina, right?
Yes.
Yeah,
so
that bottom
they
of
have some
Brandon's
stuff
looks
on the
way
bottom.
different.
It looks a fair amount different, but there is. This one looks more yellow than peachy, though. Brandon, yours looks more yellow than peachy, really. The
Just put
cap.
an observation link in the chat from New Jersey that I think has a pretty good shot of the base. See if we agree that this actually is persicina.
Hmm. I got just tapers right in.
Yeah. Yep.
But these are interesting because look at the nicely formed rings they have. Because usually the ring drops off this species, but these are probably fairly young ones that just expanded.
Give us one photo.
It's a pretty good photo, though.
Yeah, Kara did a good job. Oh, yeah, that matches Mushroom Expert. Back to Brandon's.
All right, cool. I'll probably end up calling this one chrysoblema then.
Yeah.
What else did oh yeah, this thing. So I found this today. Haven't done any work on it. I'll have it laid out for spores right now. I don't really know what family to even call this thing. Part of me wants to call it a Inocybacea. You can see they're really small, right? Smaller than a nickel. These were definitely found in moss, like in a little patch of moss. There's 2 mushrooms there and an acorn at the top, and they look very similar.
So you have a spore print color for these?
Not yet. No, I just
Yeah.
found them today. I just
They
have
remind
them
me
laid out.
somewhat of Tubaria.
That's a yeah. That okay.
I mean, it seems early for Tubaria. They come out in the winter and the early spring. But I don't know.
Cold at night. I've had
Yeah,
a few frosts.
I was thinking this is probably going to be more of a brown sport thing. But yeah, that would, Tubaria is a good lead.
Yeah, Tubaria has a print that's, I think, leans more toward yellow than brown. It's kind of like ochre usually for Tubarias.
It's got a hint of brown, but it's not like a rusty brown or purplish brown or anything like that.
And the spores under the microscope are almost hyaline, even though they're pigmented.
And would be
usually they're kind of weirdly shaped, Tubaria spores. Sometimes like asymmetrical, like a little wider on one end, and a lot of times with like guttural. Also, Tubaria have a lot of Chilocystidia that you can usually find without a lot of problems. At least Tubaria furfuracea group, it's a group. They have like Chilocystidia that are flexuous, they call it. They're kind of like
would Tubaria furfuracea group, any of those be this small in maturity?
Oh yeah, they're small. Yeah, they're small mushrooms. Yeah, if you find one that's like 2 inches wide, it's a monster.
Right, but these are mature and they're less than a centimeter.
That's pretty small. So
They're
that is pretty small.
Did you see the photo with the nickel?
Yeah, actually I did. Yeah.
They're
pretty
small. And that cap to stem Q, right? How big is that cap to how tall is the thing? Is right around 1 for this thing.
Yeah.
And in my experience, Tubaria, it's going to have a longer stem than it does a cap.
I'm not sure there's a reliable statistic there. But it seems kind of early for Tubaria.
Yeah, they do seem to have long stems.
That species does.
Yeah, all 3 of them that I looked at all have long
I looked at
stems.
Furfuracea.
Yes.
Oh.
Come on.
Now, Mycoquebec probably has Furfuracea also. See, those the stems aren't real long.
They're not short.
No, they're not real short either. It's probably not. It's probably look at this one though. This picture. This is not Brandon's mushroom here, is it?
It is.
Or it is.
The stem, it looks as big as long as the cap. No longer than that.
Yeah.
Where's the one that see a short?
Got a fairly perfect row of short gills, and then another row of even shorter.
Yeah. The one thing I will say in support of Tubaria, if we can go back, maybe you can see a little bit of the vellum on the cap.
Yeah, usually along the margin.
Familiar with the word vellum? What is that?
Velar material, so veil material, UV.
Oh, veil. Okay.
I think there's
On the
probably,
margins?
yeah, I think there's probably one photo that shows it. I think I included that. Yeah, you can see a little bit on the on either side here.
Yeah.
I suppose you can't completely rule out Cortinarius. There are some really small ones, but I don't see any signs of a cortina being deposited. I don't know.
So a way to spore print, right? At least a spore print. Get the color.
Can we just cycle through the photos? Have we seen the final photo yet? There
That's
we
it.
go. Yep,
Oh,
that's the one
that's
we
the
were
one.
looking for. Yeah, with the velar material at the edge of the margin.
Oh, that's cool. How it's sticking to it there.
The thing in the
So
upper right.
Oh,
there is an Inocybacea species called Inocybacea tubarioides because
Yeah,
it looks like Tubaria. Usually grows on. It's Brandon Matheny told me directly that it is mycorrhizal because I had asked him during a Zoom meeting because it grows on like woody debris. Um, but I mean that one has a lot of cystidia, and I think it might have nodulous spores as well. So if that's what this is, you should be able to tell without a lot of trouble with your microscope.
nice. Yeah, Tubarioides has a really textured cap, right? It's kind of got like,
Oh yeah, I believe it does. Yeah,
yeah.
not smooth like this. Yeah, okay.
But yeah, my thought is an Ossibaceae, one of those, you know, one of those genera. But
Yeah,
again, probably need more. This is a good example of need more information, right?
I think so. Yeah.
All right, cool. Thanks, guys.
Next week. Okay, thank you, Brandon. Okay, on to you, Dave. Unless anybody else wants to go.
Oh, yeah. If anybody wants to go, just go ahead.
It's not. Well, will probably be time after Dave anyway, if you want to jump in later.
Yeah, sure. Yeah, I can jump in now. No problem.
Yeah. Oh, boy, this looks familiar.
Okay, so here we are. The same thing Hunter showed. But this is interesting. We found like 4 or 5 of them growing out of white pine cones like this. So it's Hygrophoropsis
Bones. Wow.
aurantiacum. Yeah, growing right out of white pine cones. There were several of them like this. This was this was a couple weeks ago, I think. What's
That-
the date on that? Yeah, 1026. Okay, so that's a little while back. There's been a lot of these, especially under white pine. There were some on we have some white pines on our property, and we had a few of them also. So, yeah, I just thought that
Yeah,
was interesting. It was growing
that's cool.
out of
That's very cool.
yeah.
Like it's not actually growing out of the pine cone, right? It's just
I think it is.
well, it's just like the debris within
Oh
the
yeah, the mycelium just crawled up onto the pine cone. But there were
right.
like 4 of them that were like this. This was not the
Yeah.
only one we found that
It's super
was
cool.
like this. So, I've been finding some things that seem to qualify as this genus Entocybe or Entocybe. Not sure how to say it. Entocybe, I guess. So under conifers, late season, often in moss, and so the spores here are what really make me think Entocybe. And so the spores are vaguely angular, and you really have to try to enlarge that to see what I mean. So they're like Antheloma spores, but not really like true Antheloma spores.
You go
Yeah,
to
see if you
show
can do that.
original
Yeah,
image.
click
you.
on show original image down there, and then click it again, the image again. It'll enlarge
Yeah,
a
I've
little
been
bit.
trying to click it here and it doesn't want to go anywhere.
Because it's too much cropped. Yeah, so it's not going to enlarge. But you can still see. Look in the lower right. You can see those spores. They look kind of almost angular. They are angular, but only vaguely so. And that's what characterizes Entocybe. And this otherwise pretty well fits this species that's on Myco-Quebec, Entocybe priscua, I guess it is. And I found something, either the same thing or something very much like this in a few different places, but under pines, always under pines and light.
Yeah, I feel like I've seen that around too. I'm glad to have a place to maybe consider. Yeah.
Yeah, and sometimes they're bigger than this also. These are kind of small right here. And the gills are um, thinly attached, so they're pretty deeply adnexed.
Oh, okay.
Now that one, the gills look like they're pretty much adnate. So I don't think I have more than one thing here. Pretty sure these are all the same.
Yeah, there's one. Oh no, here's two. You had two to start with.
Yeah, I guess. Yeah, I guess it's really
Yeah.
these photos are all of these two. Yeah, so the gills are more deeply adnexed, and the other, and that one's not. See, that one's not very well expanded. So this one's more expanded. You can see the gills are actually kind of adnate. So
So is this something new that Baroni came up with in the last couple of years?
yeah, it looks like it's Baroni thing, and
Yeah.
Inat wants to call these things Nitida. And Nitida it's like a subgenus of Entomoma, I think, and it covers a lot of ground. And I don't know. I think INAT's just wrong. That they need to find out how to incorporate Entocybe. But I don't know.
When you say they're trying to call it Nitida, is that the genus, or they're trying to
It's
call
a
it?
Nitida. Sometimes it's hard to tell on INAT because sometimes they'll just be like one word,
Yeah,
but that one word represents like a subgenus or something, or
yeah,
a
I got
section.
that with gosh, well, it was an entoloma. Oh gosh, but yeah, someone came in and ID'd it as just the species name that I used. God, what
Yeah,
was it? But yeah, I think
maybe
you're right. I think
lavendula or citrina.
no, it was an
Or
entoloma. It was another entoloma.
and it's love. Oh yeah. Oh, leptonia maybe, because leptonia
No,
actually is a word that's been some mycologists use it to reference a genus name. And I think Nitida might be like that too. I don't know.
I
But
noticed yesterday that iNaturalist is for the sections of Amanita. They're just putting the section name. It's not Amanita
yeah, they
section
just put
something.
the section name. It's
Yeah,
confusing.
which threw me.
Yeah,
So why
it's
is it coming up that
confusing.
way?
It's confusing. Um, because INAT is not really set up for the sort of taxonomic diversity that you run into with fungi. It's just not. I mean, INAT has its good points, but quite frankly, for discussion of fungal taxonomy, Mushroom Observer is a lot better.
Anything else to say about this? Does it have what spore print color does this have? Just Entocybe? Is it brown?
I think it's got like a real pale pinkish sort of print, not quite as deeply salmon pink as true entelomas.
I don't see
I
that in.
think this print might have been pretty thin though, so I didn't get or did I put something there?
I don't see anything.
No, I guess I probably couldn't tell.
Okay.
But I just scoped another one of these, I think another one of these today that was found on Sunday. And that one was lying on the microscope slide for a long time, like almost 2 days, and that you can see that this print was kind of pink on that one, and I think it's probably the same thing as these, and that one was bigger,
Do we have
but
any
otherwise
habitat for this? We don't have any.
under pine in
Just
needles, in
okay.
moss. I think Michael Quebec says for Entocybe priscua. I think it says in pine needles. I think it says.
Anything else to say on this one?
No, just an
Okay.
interesting thing, I
You can
think
find a
I'm on the
small.
right track with
Would you
this.
call this brown or you used to call this gray?
I'd call it gray,
Mhm.
brownish disc. Looks like it wants to fade.
Okay.
and just one quick note: we don't have to go back to the observation.
Yeah.
We don't have to, but that entoloma that I was thinking of, I tagged Nick Schwartz, who knows these things.
Oh,
It was a thing that I suspected is Entoloma formosum,
yeah.
and he confirmed the ID by elevating the name to Formosa, just Formosa, not Entoloma formosum, just Formosa. So I think that's a subsection.
Oh, okay. So on INAT, they're calling. Oh, so he's saying it's probably a complex that constitutes a section or something.
Right, right.
Yeah, I
I
wish they would use these words on INAT. They just they're not
Yeah.
set up to use these words, and
And
it would be good if they did.
the only reason I know he's referring to a subsection is from a comment because I tagged him.
Right. Right.
You know.
Yeah.
However, it's
Yeah, so
quite typical. However, it's quite typical of subsection Formosa. You know.
So on
Right.
iNaturalist, if you go to the taxonomy for Entoloma formosa, they've got the genus Entoloma, then the subgenus Cyanula, then the section Fuliginosa, and then the subsection Formosa.
Oh, you can
Before
find
you
all
get to
that.
before you get to the species Formosa.
Oh,
Right.
you can find all that stuff on iNet. I didn't know that.
Yeah,
You have to
Yeah.
click on that. You know.
taxonomy tab.
Yeah, the taxonomy tab. Right.
Oh, where's that? I don't think I found that yet.
I am mostly using it on my phone, so I'm using the app.
Yeah,
And the way
it's a lot
you
easier
do
to
it,
find on the
Oh,
computer.
well, the way it's actually easy to find on the app. You just go to that
yeah, I'll look for it. Here's a I know it's
Breaking
there.
up, Dave.
I can
We're
probably
losing Dave.
find it after a while. There's really a lot of so it makes it hard to find stuff sometimes, but I'll try to find it. I'm ready to go to microplets. Probably did. Wait, when was found the tree 6? Did put these in sequencing, hopefully. Oh, this is a good one. This Crepidotus. So this was found by a Wyoming Valley Mushroom Club member at our foray on the 26th, which is a week ago from Sunday. And it's a Crepidotus. It's pretty easy to tell it's Crepidotus. You can see it doesn't have much of a stalk. It's got, looks like a little oyster mushroom. It's got gills that are turning brown. And the thing that's interesting about this one is the spores are really big. So there is a species of Crepidotus called Crepidotus fusisporus, and the spores are supposedly exceed 10 microns by a bit, but the spores are also supposed to be fusiform, which means they're pointed on both ends. And these are not so much pointed. And they're really big spores. Some of these spores are 12 microns long, or even maybe a little longer. And so I thought I had made a mistake at first. I thought, well, there was a lot of material to look at coming that I brought back home from this foray. Maybe I mislabeled one of the things that I put onto a slide. And so I dehydrated the fruit bodies, like 4 of them, and I took one of the really small ones and chopped off a little piece of gill and rehydrated in rubbing alcohol. Actually, that's what Gene Lodge taught me to do. The rubbing alcohol evaporates really quickly, so you can mount your material within 5 or 10 minutes of rehydrating. Anyway, I did that and I got the same thing. I got the same kind of spores, so I did not make a mistake. This is just some sort of Crepidotus that has really large spores, which to me seems really interesting. So I'm definitely going to send this one in.
What made you think it was special?
The large spores.
Oh, but you mean you would normally take home an ordinary looking Crepidotus?
Oh, I just take them all home. Oh, well, no. Crepidotus is
Yeah,
an
sure.
interesting genus. I have a whole book on Crepidotus.
Yeah, I know there is a book on Crepidotus. It's not
Yeah.
even available online for free. But I just wondering, I would have just assumed that this was Eplanatum.
Yeah. When you start looking at them, you
I mean, I
find
don't see the others.
lot of diversity in spore morphology, and there's there really are a lot of species. Some of these monographs that came out in the early 60s and 50s, I think maybe are kind of overdone. Like maybe there's more species than there needs to be, but
Yeah.
that's hopefully what we straighten out
Yeah,
to some
but
extent with DNA.
there's certainly more than just Applanatum.
There's a
Yeah,
lot more.
I just
There's
do
a lot.
like the three of them.
Can we
Based on the cap.
actually look
Now-
at the mushroom?
Yeah, Applanatus also typically fruits on larger pieces of wood, like bigger logs and tree
No,
trunks
I
and things
usually
like
find
that.
it on well, like I said, any kind of size, but not as small as this, perhaps. I don't know how big this one is. This one looks like a fairly small stick.
Yeah, it's pretty thin.
Yeah.
It's like an inch thick, maybe. Maybe a little more than an inch, about an inch thick. So Fusosporus supposedly grows on small sticks. So I think I have in my ID proposals, I think I said like Fusosporus could be. I think I have that down there. Pretty interesting though, I thought, because of these large spores. So Crepidotus turns out to be kind of an interesting genus. Even though the fruit bodies themselves look kind of boring.
Can we
No wonder you have a full plate if you take home sticks like this.
can we look at the photos
Spores?
maybe? No, the photos. I'd like to see the actual mushroom. Start with the second one that you skipped. The one before this.
Oh, this one?
Yep.
Stick. I can't get there. Oh. This thing keeps jumping.
Any texture on the cap, Dave?
No, that was pretty much smooth. It wasn't like one of these crocodilus or whatever. It wasn't in that. It didn't look like one of those. Yeah, I mean, it does look sort of like Applanatus, but Applanatus usually doesn't. It's usually more laterally
Like a
connected.
shells. Yeah, like a
Yeah,
shelf.
shelves. Yeah.
And what was the size on these? Oh, there's the penny. Yeah.
Yeah,
So
they're small.
Small.
The biggest one is like over a centimeter, I guess, but not a lot over that. Maybe about 1.6 centimeters, something like that.
And Applanatus can get. I think it's probably one of the bigger in the
Yeah.
genus.
That's why it's in the field guides. It's common and it's also big enough that people notice it.
I mean, I'm looking for a cap size, but the cap size, I don't see them. Is that because they're mostly, I mean, this one looks like it would have had a cap if we saw the other side of the stick. Again,
No,
it's very
these
small.
things are just centrally attached for the most part.
Okay.
Or maybe somewhat laterally attached, but I mean, they kind of remind me a little bit of Resupinatus,
Yeah.
which is not, I don't think that's what these are.
Cool. I hope you get a sequence from that one.
Yeah, I'm really hoping this works on this one. I'm saving one fruit body as well in case it's something really interesting. Maybe we'll try to get it stored someplace. So I was hoping Igor would be here. Igor, are you here?
He was on in the very, very beginning. I saw him come on.
So I'm trying to find Suillus granulatus. And this is a candidate. Because it was not growing. It did not appear to be growing with white pine. There was a white pine in the vicinity, but pretty far away. Not a real big white pine, maybe 25 feet high, and we're talking, well over 100 feet away from this. And so this was fruiting mainly under pitch pines, I believe. Did I put that in here? Pitch pine, yeah. And so I think maybe this is granulatus. I know it could be something else though. And it's certainly a Suillus.
Staining from being cut.
Just a little bit of yellowing on the flesh. I don't know if I'd call that a stain. But there's no bluing or anything like that.
Which part is supposed to have the granulatus?
The stalk, it's kind of chewed up, but there's one you can see on the apex. It does have the glands, the granules, I guess is, what somebody thought at one point. You might be able to enlarge that one. Although this is cropped, so I don't know. It might not enlarge really well. Oh, there we go.
Hmm.
Yeah, the top of the stalk. But I don't know. It's partly interesting to me because it doesn't look like the other candidates for genus Suillus that I'd expected to find in this kind of habitat. There were a whole lot of brevities in this area. There were also some acidus. I believe I tasted this one. It did not taste acidic, and there's no sign of a partial veil. Acidus has a partial veil. So, and this is not brevities.
So I've got a note from Igor next to Suillus granulatus that reads, does not occur in New Jersey. Specimens should refer to weaverae.
Yeah, it's probably an old comment. Igor has actually found something right near where he lives that he had sequenced, and it came back as granulatus.
Okay.
Yeah, so I believe that his opinion in that matter has changed. That it might at least might occur in New Jersey, and because ordinarily I would have just said this was weaverae as well, because that's what the status quo was for a while, is that all these things we were calling granulatus for many, many, many years, it should be weaverae. But then people started actually finding via sequencing that there are granulatus in North America, mostly, if not exclusively, under 2 or 3 needle pines, hard pines. But Igor has one that he found on his property that he had sequenced that I believe that, you have to ask him, but I'm pretty sure I'm quoting this correctly, that the only reasonable associate is a white pine near the complex where he lives. So that would be very interesting.
Dave, did you say this was pitch pine though, right?
Yeah, this is pitch pine. There was a white pine, 1 or 2 white pines in the area, but way too far away to have any impact on what this was associating with. So probably pitch pine. So yeah, so that's one reason why I want to see, is this the true granulatus? So I'm going to send in a sample of this.
Yeah, because my only other thought was, and I don't know Suillus all that well, but Hertellus is also
right? Yeah, I know. I thought about Hertellus also.
Also a white pine or hard pine associated.
Yeah, that's a hard pine associated. I think that's when you get in the pine barrens, right?
Yeah. Yep.
Yeah.
Well,
So yeah, I thought about that also. I think I looked at photos and read descriptions, and I forget what it was. There was something that didn't seem to really key out, but yeah, I thought about Hertellus. So I mean, I've got it listed as granular. I don't even think I have Hirtellus proposed, but maybe I should have put it in as a could be at least. But yeah, I'm going to send this in.
They're equally as reported with hard pines, right? So I mean,
Yeah.
either way, this is a cool find with a pitch pine.
Yeah. 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 I'm going to send it in. It was an interesting find. Oh, and this thing here. So a guy who was with me, he's just joined our club. This guy, he's like a well-known naturalist around here. He's real good at with like bugs and amphibians and fish and trees and flowers, but he wants to learn more about mushrooms, so he joined our club. And he put this on INAT and ended up with the proposal of Cortinarius. And I think that's what INAT suggested. And to me, I just couldn't figure out what this was. And finally, I got around to looking at spores, and the spores do look like possibly Cortinarius spores, although I see no sort of evidence of a Cortina. So I don't know. These are kind of interesting. They are going to go for sequencing, hopefully. But these spores do not look unlike what, Cortinarius spores are very, they're like the genus. The genus is very diverse. The spore morphology is very diverse. But there are some species that have these like large, fairly large spores that are kind of pointed on one end. So
that one kind of at the let's call it 130 kind of in more the middle of the screen looks pretty varicose. Like it looks like it's got some
you can't
boards.
really tell with this scope because the resolution's not great. It's good enough to get dimension estimates, but not good enough to assess ornamentation. But I may have looked at these through my other scope and wrote something in the notes. See if there's something written in the notes. No, I guess I didn't put anything in the notes. But I believe I put a proposal in here. It could be a Cortinarius.
You did.
Yeah, so it's gonna go in. It'll go in. Hopefully it'll get sequenced. I believe these were under white pine, but I'm not sure because it was a club foray and people kind of spread out. Most of the members stayed around this stand of white pine, big mature white pine, and I think that's probably where these were found, but I'm not sure because people went all around and brought stuff back and didn't get a good record of where things were found. But it looks like this was probably growing from soil at least. So, the bottoms of the stalks look like they're pretty well caked in soil.
Yeah. Can we go back to right your proposal of Cortinarius on the main page? Did you have some notes there?
No, oh yeah, what did I say? Pigmented spores. Oh, no, I didn't find any cystidia on the gills. Most Cortinarius lack prominent hymeneal cystidia. So not found something is it supports the proposal.
Yeah, no.
Let's say, for instance, if it was a Satharella, usually you can find some prominent hymeneal cystidia on Satharella spores. Just as a for instance.
Thank you, Dave.
Oh, you're welcome.
Yeah, I wonder what that turns out to be.
Yeah, well, if I find out, in a year or whatever. I'll try to remember to let you know.
Yeah, bring it back.
That was an interesting one. It was the fruit bodies are in pretty good shape. The photos are not too bad. So yeah, that's a good one.
Does anybody else have anything they'd like to share?
I've got some to share.
Okay. You share from your screen. I'll stop my share.
Yeah. All right, so first up is a lichen from the Estolmanophore. This is
You want to look at lichens?
cyanolichen in the genus Peltigra.
Peltigra.
And this one's kind of an unusual one because the upper surface has no tomentum on it. So a lot of the more common Peltigra in New Jersey, Pretextata and Canina and rufescens all have white tomentum around the edges, the lobes on the top. But when you come across one that has no tomentum, it's usually something a little bit more interesting. So this one, I believe, is Neopolydactyla, but there's not a lot of information on it. And I think the taxonomy of some of these Peltigras needs work. But this is a neat one, definitely different than what I typically see in New Jersey.
That is why, yeah.
So the orangish red is the fertile surface at the very least.
Yeah, they're the Apothecia.
Apothecia. Cool. Thank you.
And see how they kind of wrap around. Sometimes they're more disc shaped, and sometimes they're more what they call saddle shaped, like a horse saddle, and they can wrap around. Although these are even more wrapped around than a lot of the times.
This thing looks like it was just on the ground.
Yeah. These things are typically terrestrial. See some moss coming up. There is some rock. This area has some like ruins, so there's some rock underneath it. So it's kind of on mossy rock, on the ground.
Thing's cool.
Yeah, super cool.
Yeah.
And second, I've got the Calostoma lutescens from the Wells Mills foray. It's just a cool species. I like it.
Oh, you got a calostoma too now. Cool. So wait, which one did you say this was?
Lutescens, the collared one. You can see the collar on this one that rules out Ravenellii. And
which was the jelly one we saw last time? What did we decide that was? The one with all the
Ravenelli.
Cinnabarinum.
Oh,
with the pomegranate seed look. Yeah.
Yeah, they're usually more red.
Okay. And this is what is it?
Lutescens.
Lutescens. Yeah.
Nice photo.
Yeah. This isn't a cell phone shot. This is my DSLR camera. All right, and then next is a Tolypocladium that Elizabeth found today in the Pine Barrens. It's one of the ones on Elaphomyces, and it's connected directly to the Elaphomyces instead of having those rhizomorphs, which would indicate Ophioglossoides.
Yeah, and often Ophioglossoides is easily the hardest one to get the Elaphomyces, the false truffle, from,
right.
because of the rhizomorphs.
Yeah.
And it also has a way different morphology. It's got a more tongue look, with a tapered. It tapers much more. It's not as capitate, if that makes sense.
Yeah,
it's
it
not
actually looks
longer than wide.
like geoglossum or trichoglossum kind of
Yeah.
little bit. And these are more distinctly capitate. This looks like the one Brandon found, actually.
Very
Look at how bumpy the cap is.
Yeah, that's
and the color of the cap too, because
Yeah.
I found them, they've been more green on top. This one's got that blue tinge too.
Well,
I
here
think
are
that's
those.
I think that look at the spore parts broken off in the
No.
foreground.
Looks like what
just
you
above
found,
that.
Brandon.
Yeah,
Yeah, these microscopy photos aren't as good as yours, but I think it's the same thing.
I think it might be. And if it is, that just goes to prove to show like these things that, yeah, call it valiform. Is it rare, or are there just two people that put these things under the microscope that know what they found as soon as they saw it? They know to get the elaphomyces and then to do the microscopy on it. So is it rare, or is it just, underobserved?
It might
Or is
be
or is
rare, a rare year. It might be
right. I was
something
gonna
that
say
comes out once every 20 years or something.
is 2025 the year of the valiform?
Ah, yeah.
Yeah,
I mean,
yeah.
it has been a weird fall, you know, because these things are typically they come out. You normally start seeing the other species, Captatum, segmentatum, coming out right around now. But it's just been a weird late fall. It's been pretty warm, right? It was 65 degrees today.
Yeah, and it's been dry, and then we got rain.
Yeah, so maybe it's just one of those weird conditional seasons that it's the year of valiform. Cool. I would say that's the same thing that I found.
Next up is penicillium on, I'm guessing it's some sort of scat. At first I thought it was some sort of insect. It's kind of crescent shaped, but it's got a definite kind of bluish hue to it.
What are you calling this?
Penicillium.
Oh,
Yeah, we saw this, someone else showed it recently.
I think Chris had it recently.
Yeah,
So that's the
and someone else too.
capitulum.
That's neat looking. That's pretty cool.
So this kind of multi-branching, I don't know what the structure is, but this kind of tree-shaped thing is something that was in the description for the INAT suggestion, which was Penicillium vulpinum.
yeah. So are these spores asexual? Or are they links of conidia spores? Because I think that structure would then be called a conidiophore.
Yeah. I think they might be conidiophores. I'm not sure.
Did you just mount this in water?
This is mounted in KOH.
Yeah, I would say the structure is a conidiophore, and that is the structure that bears the conidiospores. And you can make out on a couple of these apexes of the conidiophore just one individual spore kind of being formed off the tip there.
Mhm. So yeah, I call it Penicillium vulpinum.
Nice. I must have missed those shares because that's a cool find.
Yeah, I just found it about 2 or 3 hours ago.
What was it growing on? It's dung, right? But
I think it's deer dung, but
oh,
it
okay.
was weird because it was just this one pellet. It wasn't like a pile, so I'm not sure.
Hmm.
I was gonna vote deer.
Yeah. All right, and then my last one is a rare lichen. This is Vulpicida Vulpicida viridis, which is proposed for the IUCN rare list and grows on Atlantic white cedar. I think I have another photo of it here. You can see the pycnidia, which are these black things on the margin, which release conidia. It's an asexual form of reproduction. So this is one that I've been looking for a while, and I finally found it today.
That's cool. Where were you?
Close to Batstone.
Nice. And is this on a living twig or?
Yeah, kind of like it's a kind of like a side branch, but it's alive.
Mhm. Cool.
That thing reminds me of like a curly kale that got whacked up by too much frost. It's got such a particular look to it.
Yeah.
And the uh, I know the word, but the vertical structures, the columns that are mixed in the center there, that's a different lichen, or is that part of this thing?
Yeah. Podetia, I think that's
Yeah,
a cladonia.
Cladonia, right? Okay,
Yeah.
Podetia.
Yeah. Actually, that looks like Cladonia didyma. Probably has a
Yeah.
red apothecia because it has squamules, big squamules going up to Podetia.
And you were talking about the black things. Is that the
Mhm.
apothecia on this?
So these are pycnidia,
Okay.
which release conidia.
Oh, gotcha. Pycnidia.
Yeah,
Gotcha.
PYC.
I guess I didn't realize that lichen had a asexual, structure
Form.
as well.
Yeah, because typically this thing has apothecia. This would benefit. This is what the apothecia of the species look like. So that's its sexual reproductive mode, and then the pycnidia here are asexual.
Okay. So will the pycnidia do not develop into the sexual
No,
thing?
the minor, from what I understand, the pycnidia are some, because they release conidial spores, they're asexual.
And it's not like
Uh,
they reach that reproductive stage and those things become the apothecia.
Yeah, I don't think so. I think those
Okay.
pycnidia are mature and they're releasing mature conidiospores.
Cool. Yeah, that's wild.
My understanding, but
And
lichen reproduction is a lot more complicated than a lot of people understand.
Yeah, I talking to Jason at the thing for like 5 minutes. It's just like, oh man, we're talking different worlds now. Um, is the color differentiation on this thing, the yellow and the green, um, is that typical, or is that environmental, or you know, cold or rain affecting that? What
Yeah,
is
it's a good
that?
question. I don't know why sometimes it's more yellow than other times. I think, this looks particularly dry, so it may be a case where when it's a little bit more moist, after it's rained, it's a little bit more colorful. I mean, most lichens do that.
And rare, has this been found, documented in Jersey before?
Yeah, there are records from the Pine Barrens scattered around. So the most recent record was 2003.
Oh wow, cool.
Yeah, that's all I got.
Thank you, Jason.
Hmm.
Does anybody else have anything they'd like to share tonight?
I just have one.
Okay.
Jason, you and Lizzie should try that Tolypocladium probably.
Yeah, I've got it. I think it should be a dehydrator drying.
I actually, those things dry pretty easily. It probably should throw it in the dehydrator, but I've gotten away with just keep it in the sun tomorrow. It's going to be like 70 degrees, a little bit of airflow, and it'll be a potato chip by the end of the day.
Cool.
Cloudy tomorrow.
All right. I found this over in Shark River. I'm not sure if anybody knows these well, but uh, thick and matted on the top. Not really torn. I got that yellowish surface. And there was a good bunch of them on this tree there.
Oh wow.
Yeah. And it's this is one that came up during the cultivation curiosity, the one on the dyer mushrooms. They said this was a dyer, and I had happened to look at it, and then a week later, here I am, like, oh, there it is.
Can't disagree with the ID. I mean, looks pretty in a notice hispidus to me, but I mean, we all we need help with the polypores for sure.
Yeah, but it was cool. It was definitely a tree full of them.
What is that and what is that tree?
Oak tree.
That's what I thought. Okay.
Yeah, but not one of the water loving oaks, right? Not like water oak or willow oak.
No, it's probably uh what are the ones with like the sharp tips on the lobes type of oak?
Red oak.
Pin oak?
Mm, yeah, maybe something like a red. but
Black. My guess is black oak. Orchis volutna.
From the bark.
From the
but they
bark,
were
yeah.
fairly
Red oak doesn't really occur in the pine. Oh, is this Pine Barrens? Yeah, sort of.
Shark River. It's got a little bit of everything.
Yeah. Usually you don't get red oak until you get up to North Jersey. And some of those actually, yeah, some of those leaves on the adjacent oak. Well, that kind of looks like
Could it be maybe a southern red with the pointy tip leaves?
do we know it has pointy tip leaves?
I mean, there's that kind of oaks in that area, but it looks like some of these other ones around it might
Go to the
be.
next photo to the right.
Yeah,
Yeah,
see
see
some of them other ones behind it.
yeah, some of those look a little bit more like Marylandica
Yeah,
Yeah, black. So is that? Would you
or
say
black.
what was the species name on that?
Marylandica. But I don't, Marylandica doesn't usually get that large. So
Wait, what's blackjack oak? Is that Marylandensis?
Marylandica.
Oh, it is. Okay.
but yeah, that's all I had.
Thank you, Chris. Anybody else before we call it a night? Okay, well, thank you, everyone, and we'll see you next week.
Thank you,
Hopefully-
Sue.
You're welcome.
Thank you, Sue.
Thanks, Sue. See you next week.
All right, all.
Good night.